Hello, my name is Victor Stolc. I'm a senior scientist at the NASA Ames Research Center. Thank you for the invitation to present our research results and hypotheses on accelerated somatic mutation and autoimmune disease. This presentation is based on a long-standing collaboration with colleagues from NASA, Stanford School of Medicine, and SETI. We have been working on the problem of understanding timekeeping in biological systems using genome science technology, high-performance computing, and next-generation sequencing in the unconventional and extreme environment of spaceflight, which includes metabolic stress in astronauts and in all studied model systems. It is now recognized that there is a need for fundamental study of somatic mutation in all human diseases because of the many varied phenotypes and genomes. The results and hypotheses presented here are somewhat surprising and even have implications for the topic of this symposium on orphan and rare diseases of autoimmunity, which I hope you may find interesting. Time is an essential non-random variable of the information content and processing functions in cellular systems. Please keep this in mind for the rest of this talk. What is somatic mutation? Somatic mutation is the gradual accumulation of mutation in the DNA of somatic cells in the body, not germline cells, that occurs over the lifetime of an individual. Somatic mutations can be point mutations, just single nucleotide changes, or can involve much larger segments of DNA, including deletions, inversions, frameshift mutations, and so on. Causes of somatic mutation include errors in DNA replication and exogenous damage to the genome, which I will talk about in a bit in my presentation. Various consequences of somatic mutations are possible. They include lethal somatic mutations, silent somatic mutations in cancer, and we can ask the question, what can we learn about somatic mutation from rare and orphan diseases? One such example is ataxia telangiectasia, also known as Lewis-Barr syndrome. It is a complex autosomal recessive disorder affecting multiple systems within the body: progressive neurological impairment, cerebellar ataxia, variable immunodeficiencies, and multiple organ manifestations. AT is a rare disorder affecting 1 in 40 to 100,000 individuals worldwide. It is caused by a mutation in the ATM gene, which coordinates the cell's response to double-stranded DNA breaks, or DNA repair, in some cases apoptosis, so that the cells with damaged DNA cannot be propagated. Individuals with AT, in addition to all of their problems, have dramatically increased rates of somatic mutations and a dramatically increased rate of cancer, especially lymphoma and leukemia. Our understanding of ataxia telangiectasia has provided physicians and scientists with tremendous insights about the link between somatic mutation and cancer. It is natural, therefore, to ask the question, are autoimmune processes also triggered by somatic mutation? Type 1 diabetes, or insulin-dependent diabetes, is less known because of the preponderance of type 2 diabetes, the non-insulin dependent, but oftentimes insulin requiring in society. It's easy to forget about type 1 diabetes, but because it is so much rarer and affects fewer than 200 cases per year in the United States. Type 1 diabetes can be viewed as a type of autoimmune disease because it involves a dysregulated inflammatory response against self-antigens, in this case, the insulin-producing islet cells in the pancreas. With this hypothesis about type 1 diabetes, the persistent proliferation of self-reactive lymphocytes that is usually hindered by a series of checkpoints is somewhat permitted. Substantial evidence suggests that proliferation of self-reactive lymphocytes is due to somatic mutations in these expanding or multiplying cells, allowing them to bypass multiple regulatory checkpoints, thereby allowing autoimmunity to take place. So the next question we like to ask is, might somatic mutation also lead to the formation of neoantigens that triggered the development of autoimmune disease as cross-reactivity to neoantigens. A broad review of autoimmune disease is a good idea before we go further, and that's shown here on this slide. Autoimmune diseases encompass over 100 conditions characterized by the immune system's misguided attack on the body's own organs, tissues, and cells. Prominent examples include the type 1 diabetes, rheumatoid arthritis, systemic lupus erythematosus, and celiac disease, among others. While the exact triggers for most autoimmune diseases remain elusive, it is widely believed that a combination of genetic factors, infections, and other environmental exposures significantly contribute to their development. Although treatments exist for many of these conditions, the cures remain elusive. Autoimmune disease imposes a heavy burden on patients, resulting in diminished quality of life and substantial medical expenses. Beyond affecting individuals directly, these conditions have a ripple effect on families and communities, emphasizing the need for better understanding of these disorders in hopes of developing prevention strategies and better treatment. Specific biomarkers such as autoantibodies, T and B cell profiles, and cytokine signatures are utilized for diagnosis and monitoring disease progression. Understanding disease-specific immune profiles aids in accurate diagnosis, disease monitoring, and personalized treatment strategies. So the next question we want to ask is, what are the origins of mistaken identity in autoimmunity? The exact origin of the mistaken identity in autoimmune conditions is not fully understood. However, there are several theories that attempt to explain the underlying causes. Here are a few examples. Genetic factors, in particular, are believed to contribute to the development of autoimmune diseases. People with a family history of autoimmune conditions are more susceptible, suggesting a genetic predisposition. These triggers can disrupt the immune system's normal functioning, leading to the recognition of self-tissues as foreign and the subsequent immune response. Other forms of mistaken identity in autoimmunity include molecular mimicry, which occurs when an infectious agent or an environmental substance has similar molecular structures to the body's own tissues, and the immune system may target these foreign substances, but in the process, it can also mistakenly attack similar-looking self-tissues, leading to the autoimmune reaction. Loss of immunity of the immune system to recognize and tolerate the body's own tissue as self and not mount an immune response against them. In autoimmune diseases, this immune tolerance is disrupted, causing the immune system to mistakenly target self-tissues. Additionally, dysregulation of the immune system, which is a kind of hand-waving hypothesis about the immune system having an imbalance in the regulation of itself. So I won't spend too much time on that one, but I think one of the most intriguing ones that can help explain many autoimmune diseases, the environmental triggers, specifically the microbiome, which is the link between the environment and the patient. Despite their microscopic size, microbes play significant roles in maintaining human health. They safeguard us against pathogens, contribute to immune system development, and aid in food digestion for energy production. The microbiome encompasses all of the microbes, including bacteria, fungi, viruses, and their genomes that reside on and within our bodies. The number of microbial cells in the body equals the number of human cells. This is a very important point to understand and to think about, because a person's microbiome is formed in the first years of their life, but can change over time in response to different factors, including diet, medications, and environmental exposures. The variations in the microbiome can lead to distinct health effects, resulting from environmental exposures and may influence an individual's susceptibility to disease and disruptions of the microbiome caused by the environment can heighten the risk of conditions such as diabetes, obesity, cardiovascular, and neurological disorders. So it is becoming increasingly clear that there is a link between metabolism and somatic mutation. If that is the case, it raises the following questions. Might some of the products of metabolism mutate the human proteome, which is somatic mutation, or might the microbiome generate antigens that are cross-reactive with self-antigens? Are foreign microbiome molecules being generated that trigger autoimmune disease? In essence, it is a chicken or egg dilemma. When we compare the human versus the microbiome proteome in terms of its potential for generating antigens, it is important to recognize, as I mentioned earlier, that representationally microbes account for the same number as our own cells within our body. While the human proteome encodes 20 to 25,000 unique proteins, the microbiome collectively encodes millions of unique proteins. Additionally, while the human genome is relatively stable and consistent across individuals with only minor genetic variations, microorganisms have much faster mutation rates. The genetic diversity translates into a larger pool of antigens that may mimic self-antigens and drive autoimmunity. While there is no specific microbial antigen that has been identified definitively for some of the rare diseases such as myalgic encephalomyelitis, several studies have explored potential links between microbial infections and the onset of exacerbation of symptoms in some individuals. Here are some examples. ME has been linked to infections with EBV virus, herpes simplex 6 virus, cytomegalovirus, enteroviruses, and other viral infections. However, a question about this is that most of these viruses actually resides in vast majority of people, and in fact, some of them, such as CMV, are so prevalent that they're not even being tested for in blood transfusions. So the question is, how do some people end up with a very rare autoimmune disease such as ME, and other people don't? This slide reviews some of the microbial antigens that share structural similarities with human antigens. Microbial antigens that share similarities can contribute to the development of autoimmune diseases through a phenomena known as molecular mimicry. Here are some examples of microbial antigens that have been implicated in molecular mimicry. In rheumatoid fever, a bacterium called Staphylococcus pyogenes is responsible for the strep throat, produces antigens that resemble proteins found in human cardiac tissue. This molecular mimicry can lead to autoimmune reactions targeting the heart, causing rheumatic fever and rheumatic heart disease. Guillain-Barré syndrome is associated with C. jejuni, a bacteria pathogen commonly associated with gastrointestinal infections that produce lipooligosaccharides that share structural similarities with gangliosides in human peripheral nerves. This molecular similarity can trigger an autoimmune response against peripheral nerves, leading to the GBS disease. Multiple sclerosis has been associated with various viruses, including the EBV virus, and this one has been linked to MS development. It is proposed that viral antigens resembling myelin proteins in the central nervous system can induce an autoimmune response in susceptible individuals. Type 1 diabetes has also been associated with a virus called Coxsackie virus B and other enteroviruses it's because the proteins in these viruses, particularly the viral capsid protein VP1, share similarities with pancreatic beta cell antigens, leading to cross-reactivity and potential autoimmunity against the insulin-producing beta cells. So now I have already mentioned that metabolic factors can alter antigens in the body and in the gut. And now I want to give you a glimpse of some of the biochemicals that may be responsible for this phenomena. So we want to consider the question, how might metabolic processes alter antigens? So reactive oxygen species serve a timekeeping function as switches of various cellular processes and physiological rhythms. The best candidates for timekeeping functions are reactive oxygen species oscillations, such as in hydroxyl radicals, which are produced as byproducts of bioenergetic metabolism. To counter excess ROS, or reactive oxygen species, cells contain antioxidant systems, including pyroredoxin enzymes, which protect against oxidative stress. Mutants with defective peroxyredoxin enzymes exhibit increased somatic mutation caused by reactive oxygen species. More about hypermutation as a consequence of gene activity during the oxidative phase of the redox cycle is shown on the next slide. Studies in yeast provide a wonderful example of how the redox cycle system is cyclical. Even though yeast don't have lungs, they consume oxygen in a cyclical manner, as shown on top left panel, much like breathing. Redox cycle generates an oscillating pattern of gene expressions, as shown in the bottom left panel. Another way to say this is that some genes are exposed or expressed in the oxidative phase, and different genes are expressed in the reductive phase. The right graph shows that genes transcribed in the oxidative phase accumulate mutations significantly more than genes transcribed in the reductive phase. The mutation frequency is plotted on the y-axis. Therefore, environmental stress that induces reactive oxygen species, such as spaceflight that we study at NASA, have the potential to mutate DNA and disrupt biorhythms. One of the most interesting points about this data is that reactive oxygen species induced by the environment enable a coupling between heredity and the environment. This is why studying life processes in space provides an outstanding opportunity to unravel the timekeeping functions and somatic mutation at a fundamental level. So ROS that induces mutations is a fundamental genetic mechanism process that affects all species, including humans. And ROS-induced mutations may have clinical relevance for astronauts subject to metabolic stress in spaceflight. That's why we study it at NASA. If nothing unusual were going on, we would have expected 1 or 2 somatic mutations during a period of our experiment on the International Space Station, which took about 40 days. In fact, we observed many thousands of mutations in each tissue that was examined in mice. By comparison, essentially no somatic mutations were detected in the ground control mice, or many fewer mutations were controlled relative to the control mice on the ground. As shown in the Venn diagram, some somatic mutations were unique to the specific tissues, some were shared, and several of the mutations identified were common to all 4 types of tissues that were analyzed. These are the hotspots of variation in the mouse genome when exposed to spaceflight. The ISS-induced mutations lead to low levels of genetic mosaicism in different tissues, and some of the non-synonymous mutations, which are the ones that change the amino acid identities, likely resulted in loss of functions in areas of physiology relevant to spaceflight. In this slide, we illustrate the relative mutations that were increased by induced RNA transcription during spaceflight on the ISS relative to ground controls. The average mutation frequency of 10 to the minus 6 per base pair on the ISS relative to the ground controls at almost 4 times 10 to the minus 7 was identified in significantly induced mouse genes, which is about 18 times higher average mutation frequency due to spaceflight, but it is not due to radiation, but rather elevated CO2 levels. In summary, somatic mutation may play a critical role in the development of autoimmune diseases via a variety of mechanisms. Rare and orphan diseases help illustrate this phenomena. Microbial proteome sequence space, its aggregate, is 100 times more complex and mutates much faster than the human proteome, and therefore represents the most likely source of autoimmune self-antigens. Antigens that may induce immune cross-reactivity with self-antigens could be derived from metabolically induced mutations, both somatic and microbial target genomes. And a high rate of somatic mutation was observed in mice that spent only 5 to 6 weeks in space aboard the International Space Station. So we know that somatic mutation can be induced environmentally substantially to change the proteome of an organism. And non-synonymous mutations, or those that affect the amino acid sequence identity in proteins, amounted to approximately 1/4 to 1/3 of all the mutations observed, and these are the mutations that count. Elevated carbon dioxide can result in generation of reactive oxygen species, which can lead to somatic mutation, especially involving guanine base changes on the International Space Station and in metabolic diseases. So the question from this summary is, might spaceflight with its associated increased somatic mutation eventually result in autoimmune diseases in astronauts? And it's a question that we should also consider for general public, not just astronauts, because somatic mutation in the microbiome is a very rich source of autoimmune antigens. So with this, I would like to conclude my presentation and thank you for your attention, and I will take any questions you may have at this point.